Professor of Soil Ecology at the University of Wyoming. Pete has a BA in Agriculture and Biology from Oklahoma State University and an MS and PhD in microecology from the University of Wyoming. After graduating, he did a postdoc at the Center for Microbial Ecology and Long-Term Ecological Research associated with Michigan State University. The postdoc was followed by a position at the National Soil Tilth Laboratory in Ames, Iowa. After several years in postdoc positions, he returned to Laramie to fill the soil ecologist position at the University of Wyoming in the Department of Renewable Resources. The foci of his research program are soil microbial ecology and restoration ecology and the interface of these two disciplines. Please help me in welcoming Dr. Pete Stahl.
Okay, it's great to be here this morning. And yeah, I really appreciated Caitlin's first talk, gave a lot of great background information. I'll go ahead and start by adding a little bit to the definition of soil. I do this to get my students thinking in soil microbiology class or soil ecology. Okay. All right. There you go. One of my favorite soil organisms there. Okay, it's great. All right. So I like to include in a definition of soil that soil is the habitat on the planet with the greatest diversity of organisms, and actually is the habitat where most of the organisms on Earth spend most of their lives, in the soil. And hopefully what I say here in the next 25 minutes will support those statements. So again, great variety of organisms that live in soil. Here's one of them, a burrowing owl. Doesn't spend a whole lot of time in the soil, definitely uses for cover and protection, but doesn't really get much food from the soil. Oops. This is one of the soil organisms I try to avoid, one of the few dangerous soil organisms. Yeah, when they come out of the hole, I take off in the other direction. But again, very important in soil, moves a lot of soil around, keeps the soil disturbed or mixed. Has an impact on the soil very different than most of the microorganisms that live there. One of these guys has quite a range. You don't find too many of these big mean creatures in one area. On the other hand, the microorganisms, there's millions of cells in each gram or teaspoon of soil and hundreds of meters, if not thousands of meters, of hyphae of the fungi, microbial fungi that live in the soil. Okay, so here's just a depiction of the variety of different types of organisms. All these are relatively small, but widely distributed in soils, except for one that almost everybody knows down in the corner here, earthworms. Most soils I've dug holes in Wyoming, soil pits, don't have earthworms. They're just too dry. Up by Yellowstone, where there's higher precipitation rates, little moisture soils, see more earthworms up that way. But generally, semi-arid soils, arid soils don't support earthworms. Around the top shows a number of soil animals. Arthropods and flagellates, amoeba. Over here on the side is a nematode, a microscopic worm. The most abundant animals on the planet are really widely distributed in soils and water. Every soil I've ever looked for these in Wyoming, they're there. If you're a sugar beet producer, you know all about nematodes. They can be a big problem as a pest on nematodes, or excuse me, on sugar beets.
Yeah.
Let's see, down below, some more nematode photos and other soil creatures from different taxa. We'll go into these in a little more detail. Starting with the bacteria, the smallest organisms in soil, the most abundant organisms in soil. These things are found in concentrations over 1 billion cells per teaspoon of soil. They're that tiny. You will not see these guys unless you get a microscope. This is a photo micrograph taken with an electron microscope at Michigan State University. These guys are extremely important in the cycling of nutrients, the transformation of soil carbon fixed by plants through photosynthesis into soil organic matter. There's lots of organisms involved, really complicated process, as Caitlin pointed out. These guys are one of the key players. Again, thousands and thousands of species in almost every soil. Speaking with Caitlin this past week, I mentioned that a Scandinavian scientist in Finland looked at the litter layer in forest soils there and estimated 10,000 different species of bacteria per gram of soil. Okay, a little bit closer look at these simple organisms, just little capsules of biochemical activity. They absorb things dissolved in the soil solution, break down organic matter into simpler forms, and create lots of biomass. These are the organisms that are responsible for converting ammonia in the soil, which is the form of nitrogen released during decomposition, into nitrate, which is the form of ammonia, or excuse me, the form of nitrogen that plants prefer to take up. So that is a critical process involved in plant nutrition. But the plants are fixing carbon, as Caitlin mentioned, that feeds most bacteria in soil. So really important reciprocal relationships. Some bacteria have flagella and swim around in the soil solution, but most of them are attached to soil particles, pieces of organic matter, and plant roots. The plant root, as Caitlin pointed out, is exuding carbon compounds, energy-rich compounds that microorganisms can use for food, energy, and nutrition. They have huge populations around these plant surfaces. This is a picture of a leguminous plant root with the rhizobia bacteria on the very tip, getting ready to invade that to start the nodule forming process. The only organisms on the planet that can convert nitrogen in the atmosphere, that N2 molecule that we're all breathing right now, it's going in and out of us, we don't get much out of it, we need the oxygen. These bacteria can convert N2 into usable forms of nitrogen. Every organism on the planet is dependent on nitrogen-fixing bacteria to do their thing so we can, well, they're doing it for their own good, but it gets passed down through food chains and we all benefit from that. The only other way nitrogen, N2 from the atmosphere, gets converted into usable forms is through lightning strikes. Okay? There's a number of plants, these leguminous plants, create little environments, places for them to live on their plant roots. Those of you growing alfalfa know all about this and have seen those nodules. They really increase the rate at which bacteria can fix nitrogen because they're feeding them and providing them with a really great environment in their roots. Really, really important. important to soil health, important to the health of the cattle grazing on those pastures. More leguminous plants in those pastures, the greater rates of nitrogen fixation will occur in the soil. Okay, again, bacteria are widely distributed in the soil. This is a photograph I took after I took a little sample of soil, threw it in a blender, stirred it up real great, threw in a stain there, took a little bit, put it on a microscope slide, looked at it, took the photograph. Millions to billions of cells in each gram of topsoil. As we go down in depth in the soil, and it gets drier and drier, especially in Wyoming, the amount of biological activity decreases. But up at the top, it is really active. Okay, a filamentous bacterium known as actinomycetes. Caitlin mentioned the importance of antibiotics produced by microorganisms. Streptomycin comes from a very common soil bacteria, Streptomycetes, a filamentous actinomycete. All right. Another filamentous bacterium. They are abundant in Wyoming soils. Here is a photograph of some petri dishes in which some different soil organisms have been inoculated. And you can see there's zones in those plates where the organisms don't grow because they're producing antibiotics, which inhibits growth of neighbors, even if they're the same species. But this is one way soil organisms maintain their territoriality. Okay. We do have some photosynthetic microorganisms on the surface of soil. Algae are pretty abundant, especially in wetter spots in the soil. But along with the plants, they are producing fixed carbon, converting CO2 in the atmosphere using sunlight into fixed carbon to form their own biomass. Okay. This illustration is to show where the organisms in soil are living, and that's in the pore space. There's actually more pore space in soil than depicted in this diagram. Soils are approximately 50% pore space, good, healthy soils. 50% mineral and organic matter, 50% pore space. It's that pore space where the organisms live, especially the smaller ones. Bigger organisms, even some of the insects, can burrow through and make their own space in soil. Others have to live in that pore space. This is tremendously important. This is one of the things that you want to manage for good soil health, having good soil structure, because that maximizes the amount of pore space, and that's where the organisms live. Compaction of soil reduces the amount of pore space and provides less habitat space or room for soil organisms. So that's one thing you really want to avoid to maintain your soil health, is soil compaction. Okay? It reduces the amount of pore space. Another thing I wanted to mention, Caitlin talked about soil organic matters being 50% carbon. That's really important. Carbon compounds make up the food for microorganisms in soil, and they are derived from plant compounds or plant biomass. What Caitlin didn't mention is in most soils, 95% of the nutrients, including carbon and things like the nitrogen, phosphorus, sulfur, potassium, are tied up in that soil organic matter. The more of that you have in your soil, The healthier your soil is going to be, the more fertile it's going to be. Tremendously important. Organisms take up nutrients from the soil, concentrate them in their biomass, just like we do. We eat all these rich foods or good foods, get all the good nutrients, concentrate them in our bodies. That soil organic matter is basically what's left over of dead organisms, all the dead organisms in soil through different types of processing. Another key to maintaining good soil health is having lots of soil organic matter, as Caitlin pointed out. Even in soils that have been fertilized, still most of the nutrients are coming from that soil organic matter. Okay, now we're moving from the bacteria, the prokaryotic, really simple organisms, talking about soil fungi. These guys are also extremely abundant in soil. And in some soils, only plant roots are more abundant than the hyphae of fungi. These guys primarily make a living from decomposing dead plant materials. All those roots Caitlin showed in that one diagram, they get decomposed by fungi. That thread-like growth form allows them to grow into solid materials like plant roots, plant stems, plant leaves. Dead logs on the forest floor, how do they get decomposed? Fungi from the soil grow up into those logs. They're almost all carbon, don't have much nitrogen at all, certainly not enough to support organisms. Fungi, because of that filamentous growth form, can transport nitrogen from the soil up into those logs they're decomposing to help them live and break down, create more biomass, decompose those things. So these guys have capabilities that the bacteria don't. Shaggy mane mushrooms. You might have seen these growing off of old cottonwood stumps that got cut down. Really tasty. Here's a not so tasty petri dish full of fungi. Yeah, just showing the variety of different fuzzy forms of these things that can be isolated in the soil. That's how we study these things. Okay? One of the reasons we still don't know as much about soil biology as we do about other things in the soil is because it turns out that probably 99% or more of the organisms, microorganisms in soil, we don't know how to grow them in a laboratory. We don't know what they look like, except if we can find them in the soil with a microscope. Okay? We just don't know what it takes to make them grow in petri dishes. Probably some other method besides petri dishes. So we've got a lot to learn about these microbes, bacteria and fungi. All right, here's another slide showing a soil dilution. I took a little sample of soil, threw it in a blender with water, put the stain in there, take samples. This sample here is less than 1/1,000,000th of a gram of soil on a microscope slide. And yet it's got that much fungal hyphae growing through it. And that particular type of fungal hyphae is associated with mycorrhizal fungi, which I'd like to talk about a little bit because they form an extremely important symbiosis with almost all plants that helps them extract nutrients and water more efficiently than they can do on their own. More fungal hyphae in the soil. This is the fruiting body of a penicillium fungus. Another one of those really important antibiotic producers. Penicillin, that fungus, was the organism from which the first antibiotic was isolated by Sir Alexander Fleming. Is that before World War II or during World War II? But yeah, penicillium, not as widely used as it was, but still quite widely used. Okay. This slide is of a lodgepole pine seedling to show how mycorrhiza work. You can see that it's growing in a dark soil. That yellow filamentous material is mostly fungal hyphae growing out from the center axis of that lodgepole pine seedling. I think this does a really good job illustrating how a mycorrhizal fungus acts as an extension of its host plant root system and allows it to extract nutrients and water from greater volume of soil than the root system alone allows. Okay? The fungus gets all its food or carbohydrate from that host plant, the lodgepole pine seedling, and almost all the water and nutrients going into that lodgepole pine seedling are being uptaken from the soil and transported to the seedling by the fungus. Okay? Sagebrush. Sagebrush roots are full of mycorrhizal fungi. Almost all grass roots. I can't think of a grass in which I haven't seen lots and lots of mycorrhizal fungi. Really important to our semi-arid prairie plants. Here's a closer look at the roots of a lodgepole pine seedling. That yellow material is the fungus growing around the exterior of the roots. Unfortunately, all the hyphae got broken off that would be growing out into the soil when I harvest that. It's fine material, and you can see a few fragments hanging off the roots, but not much. I've got a better slide of that. Here's a sagebrush root segment that I collected. And, well, actually, I grew this one in the greenhouse so I could harvest it really carefully. I grew it in a soil with a lot of sand that made it easier to harvest. But you can see all that filamentous material growing out from the sagebrush root. That's all the mycelia of the mycorrhizal fungus, which is also growing through the center in the cortical tissue of that root segment. In this next slide, we're going to be taking a microscopic view at the inside of that root that I've stained with a blue stain that gets absorbed by the fungus. And you can see the hyphae growing linearly along through the cortical cells. It grows inside and outside of the cells. The darker line above is the root cortex, the stele, the vascular tissue in the center of the root. And then we're down just below that where the fungus is colonizing the cortex. That's almost like a little root system inside the cortical cells where the nutrients and water are released into the plant, and the fungus collects sugars provided by the host plant. Okay, again, to maintain the populations of those mycorrhizal fungi in the soil, you just want to have a good, healthy plant population above ground. That's so important to all of these soil organisms. The plants are the primary producers. They're fixing CO2 using sunlight, making all these organic compounds in plant biomass. Eventually, that all gets down and used and recycled by soil microorganisms. Another really important feature of all that fungal hyphae in soil, and when I worked in Iowa, I studied soils there, a lot more moisture, more plant production. We found that uncultivated tall grass prairie soils in Iowa had over 1,000 meters of hyphae in each gram of soil. If you extrapolate that a little bit, a square meter 6 inches deep has 25,000 miles of hyphae. Okay? A lot of that is holding soil particles together, facilitating and maintaining soil structure, almost like a little biological rebar system in the soil, okay? Holding soil structure together. Okay, another